The reversion of induced head shapes in flatworms offers a rich context for exploring the mechanisms of morphological stability and plasticity. The tendency of the organism to return to its default state can be understood through the cell-M framework. DNA encodes morphological potential. The cell-M suggests that DNA contains the necessary information to specify anatomy, with cells interpreting this information probabilistically. Environmental inputs modulate outcomes. Bioelectric signals and the microenvironment can temporarily alter morphological expressions, but the cell-M DNA guides long-term stability. Entropy in biological systems. Entropy may influence biological systems differently than in physical systems, with living organisms actively maintaining order through energy input and regulatory networks. The proposed experiments to investigate the role of external fields in shielding could provide valuable data to test these concepts. Proposed experiments: shielding and field exposure. Experiment 1. Shielding flatworms to block entropic forces. The objective here is to determine if shielding flatworms from external influences slows down the reversion to the normal head shape. So, let's talk about the methods. We will place induced flatworms in a Faraday cage or other shielding apparatus to block electromagnetic fields. And of course, we will include control groups with flatworms in standard conditions for comparison. Now, what are the expected outcomes? If external fields influence reversion, the shielded flatworms may show delayed or altered reversion. On the other hand, if the reversion is intrinsic, there should be no significant difference between the shielded and control groups. Experiment 2. Exposing flatworms to different levels of radiation in fields. The objective here is to observe how exposure to radiation, electric and magnetic fields affects the rate or nature of reversion. Let's dive into the methods. We will subject induced flatworms to varying intensities and types of fields separately. Monitoring will be crucial. We'll track morphological changes over time compared to control groups. And what do we expect? If there's a modulation of reversion, changes in reversion rates or patterns could indicate that external fields influence morphological stability. But if reversion is governed by internal mechanisms, external fields may have minimal impact. Implications of experiment results. Influence of external factors. Positive results would suggest that environmental fields play a role in maintaining or disrupting morphological states. Intrinsic versus extrinsic control. Findings could shed light on the balance between internal genetic programming and external environmental influences. The cell-M theory and transient morphological changes. Cell-M explanation for reversion. Evolutionary training data. DNA contains the evolutionary training data for the species, encoded in the resonant connections within the genome. Local environmental adaptation. Cells interpret environmental signals through the cell-M, producing probabilistic outputs that guide development. Unsupported changes. The induced head shape is not in a stable state within the cell-LM's encoded possibilities for that environment. However, it will provide a probabilistic output for the altered environmental inputs, that is, bioelectric cues in the microenvironment, allowing the cell-LM to revert to a probabilistic output for the supplied inputs, generating a historical adaptation to the environment based on learned evolutionary data. Environmental inputs and morphological outcomes. Bioelectric cues. Changing bioelectric potentials alters the microenvironment, leading cells to produce different morphological outcomes temporarily. Reversion driven by cell-M. Once the external perturbation is removed and the chain reaction is complete, the cell-LM guides cells back to the default anatomical pattern currently encoded in DNA as the default structure. Implications for morphological plasticity. Transient adaptations. Cells can temporarily adopt different states in response to environmental changes, but the cell-LM ensures stability over time. Role of bioelectric fields. Bioelectric signals modulate the resonant connections, influencing the probabilistic outputs without altering the underlying genetic code. Integration and future directions. Understanding morphological stability. Balance of forces. Morphological outcomes result from the interplay between genetic encoding or cell-LM, environmental inputs, and physical forces including entropy. Regenerative dynamics. The reversion to the original head shape illustrates the robustness of the developmental program encoded within the cell-M. Exploring environmental influences. External fields as modulators. Investigating how electric and magnetic fields affect morphological stability could provide insights into bioelectric regulation, entropy in biological contexts. Further research is needed to understand how entropy interacts with biological information processing systems like the cell-M.